Thank you everyone for joining. We have the incredible Elli Brimble with us, who was a genetics counselor at Stanford and has moved over to Ciitizen and is leading our natural history study along with how many other disease groups is it at Ciitizen?
Quite a few. I think we're around 10 and we're hoping to support another 15 this year. So
it's incredible. And for those who don't know, Elli and Nasha really spearheaded the grant to the Chan Zuckerberg Initiative to reinvent the way natural history studies are done. I mean, it's so incredibly innovative. Many of you might know that in the past, natural history studies were done by bringing your child into a clinic once or twice a year, and they would observe the child, and that would be the data they would get for the natural history of the disease. And Elli has spearheaded this incredible grant with the Chan Zuckerberg Initiative to get 1,000 times more data by using machine learning to extract from our children's medical records. Because really all the information about what our kids go through is in their medical records. So it's brilliant, and we're so lucky to have you on board. So without further ado, I will send it over to you, Elli.
yeah, and we've been working on going through some of the data that we've collected. And so I'm going to be really excited to share that with everyone in the new year. But one of my favorite statistics that we've looked at so far is just through participating from the first 50 participants, we've collected over 350 years worth of data. So it's just an incredible amount of information that we're really excited to be able to use to spearhead new treatments and just a better understanding of this condition. So certainly grateful to be welcomed into this community and for everyone's participation.
Yeah, and a lot of people here, well, everyone here is in the natural history study. So thank you all, because we're really this is what it's going to take to get treatments for our kids and for the future. So we're really at the beginning of something really important.
Yeah. So today we're going to talk a little bit about FOXG1 genetics, and I put together a slide deck to go over some of the introductory concepts to genetics. And then the goal is to reserve some time at the end to answer any questions that people might have about specific genetic reports. One thing that I did want to let everyone know is that we are talking about genes and specific mutations, which is private information. So if anyone has questions that they'd like to discuss one-on-one, we're going to try and save some time at the end for some one-on-one conversations as well. And I'm always happy to reach out by email after. But for those who are comfortable sharing in the meeting, we're happy to support that as well. So to get us started, I'm going to talk a little bit about genes and proteins. So these are words that we talk about a lot in this community, but maybe haven't actually sat down to really define. And so I wanted to do that here. So when we talk about a gene, we're really just thinking about a section of DNA that codes for an instruction in the body. And we can think of each gene as a string of chemical letters. And there are 4 of these chemical letters that are repeated in different combinations. And they're A, G, T, and C. So if you've ever looked at your genetics report, you may notice that when they're talking about DNA, they're only going to be using these 4 letters. Like any sentence or book that we're reading, the sequence of those chemical letters are important to make sure that the stored instruction is accurate. So small typos to the spelling of those instructions might change the meaning or the function. Most genes that we talk about store instructions for a protein. And proteins are molecules that perform important jobs in our body. So they might do things like make chemical reactions. They can act as signals between different parts of the cell or different parts of the body. And they also can form important structures in our body too, among many, many other functions. So when we talk about FOXG1 is both a gene and a protein. And the FOXG1 gene stores the instructions for creating FOXG1 protein. There are more than 20,000 characterized proteins in the human body, and unsurprisingly, given its complexity, many of those are expressed in the brain, and FOXG1 is one of those proteins that primarily works inside of our brains. When we talk about different conditions, some can be caused by changes to the spelling of our genes, which is like what we see in FOXG1 syndrome. So if we look at this example here, we have this sequence of DNA letters. And if we look at this letter here, we see that the C has been changed into a letter G. So it's a very, very small change, but we know that it might change the meaning of the instruction. You might hear these different types of changes called variants or mutations. I tend to use variants because it doesn't necessarily have like a positive or a negative meaning. It's just a change found in our genes. There are a lot of different types of changes that can happen in our genes. So we just looked at one where there was just one small typo, but sometimes there might be changes where a letter is missing, or maybe more than one letter is missing and there's a chunk of information that has been deleted. Also, sometimes there might be information inserted or duplicated. So here we see an extra C has been added into the sequence. All of these are different types of genetic variants, and we see all of these types of changes in FOXG1 syndrome. FOXG1 syndrome is what we call an autosomal dominant disorder. And importantly, most variants are not found to be present in a parent, meaning that these are brand new changes that occur either in a sperm or an egg cell or very early on in development of an embryo. And then just as a reminder, we have 2 copies of every gene, 1 copy from the biological mother and 1 copy from the biological father. And so every gene in our body, including FOXG1, we have 2 of those. And in autosomal dominant disorders like FOXG1 syndrome, there is only one FOXG1 mutation that impacts its function. So one copy is perfectly healthy and the other copy carries a genetic variant. And we know that that single copy alone that single variant is enough to cause symptoms. Even if the other copy is healthy, having one variant, FOXG1 is an important enough of a gene that one change can cause the symptoms that we see. When we talk about FOXG1 often being a de novo condition, what that means is that the variants that we identify in our children are typically not inherited. And so like we talked about earlier, de novo variants typically occur in either a sperm or an egg cell or very, very early in development, which makes it tricky because we can't always predict if someone will have FOXG1 syndrome or not. It's not something that we can necessarily test parents for in advance. So I'm going to switch gears a little bit to talk about dissecting a genetic test report. So to make things extra confusing, every lab has their own format, but for the most part, labs report the same types of information in a somewhat standard way. So this is an example from a genetic test report, and we're going to work from the left over to the right. And so most reports are going to highlight the gene in some fashion, and it tends to be the first thing that they talk about. So we see here in the first column of the table that the gene is FOXG1. And so this is going to be the gene where a genetic variant was identified through the testing. The next sections are going to talk about the specific coordinates of the genetic variant. So where is the change located and what was the change that was found? Some labs report these 2 findings out together and other labs break them out like we see here. And basically, these are just 2 different ways of talking about the same information. So in the second column, we're looking at the change that was found in the DNA sequence, which in this case says at position 670 of the FOXG1 DNA sequence, the letter G has been changed to a letter A. So it's a very small spelling typo. And what this results in the corresponding FOXG1 protein is a change in the protein sequence. So where we would expect to see a glycine amino acid, which is one of the building blocks of protein, the change in the DNA sequence resulted in a corresponding change in the protein, where now we see a different amino acid called serine. So this is kind of like a lot of terminology, but really what we're describing here are small changes in genetic variants that result in symptoms of a condition. So this area is really just describing the type of change that was found. A lot of labs will also comment on the zygosity of a change. So remember a couple of slides ago, we were talking about how we have 2 copies of every gene. And so reports that say heterozygous mean that there is only 1 copy identified. So in this report, we're saying that only 1 copy carried this variant. Whereas you may see in other genes that the zygosity might say something like homozygous. And that would indicate that both copies of the gene were found to carry this variant. So in FOXG1 syndrome, we're really only going to be seeing something called heterozygous changes. And then the last column in this table, which is in some ways one of the most important columns, is the lab's interpretation of the finding. So how confident are they that this genetic variant that they identified is relevant or clinically meaningful? So we all have genetic variants, and many of those variants are completely benign, but they just contribute to making us all, our unique selves and different from one another. So one of the responsibilities of the labs and the clinical teams that you may work with is to take this information and make a prediction about whether or not we think this is causing the person's symptoms. So do they have enough evidence to say that, yes, this FOXG1 variant explains the person's clinical symptoms and means that they have FOXG1 syndrome? So a lot of times we're going to see things like pathogenic or likely pathogenic, which means that the lab is very confident that the change is going to impact FOXG1 syndrome or FOXG1 function. And so they're very confident that the person likely has FOXG1 syndrome. And other times we might see something called a variant of uncertain significance. And that just means that they wanted to show us the variant because it is likely not present in many people who are sort of healthy and don't have symptoms of FOXG1 syndrome, but they didn't quite have enough evidence to say for sure that that is the cause. And a lot of times this happens when there's a variant that has never been seen before. And so we need a little bit more help to understand what the impact of that variant is. All right, so we talked about this. And then, one thing that you've probably heard us talk about over the years is that we're really interested in classifying different variants of FOXG1 syndrome by their kind of categories of mutation types. And so I've listed some different mutation types here. We know that one of the most common types of mutations in FOXG1 syndrome are frame shift variants. And so this occurs when there is a small piece of information missing or added, but it changes the reading frame of the protein. And so what that means is that where we might typically say that these 3 letters together form a word of CCT, if we make an uneven change, it changes the meaning of the word. So the body would essentially skip this blank and say CT and whatever the next letter is. So it's not only that something is missing, it's actually changing the entire sentence structure of this genetic information. And so again, that's kind of a lot of mumbo jumbo, but what it actually means is that rather than just one single change, a lot of the FOXG1 protein is going to be changed as a result of this. And most of the time, what happens in a frame shift is it ends the protein early. So early, there's going to be missing information as well as changed information. Another very common type
Elli, is that called an early truncation?
Yes.
Okay.
So sometimes you may hear us talk about truncating variants. And so things like frame shift and nonsense variants are truncating because they shorten the sequence. It's the FOXG1 genetic instruction is shorter than we would expect it to be compared to someone with a typical FOXG1 sequence. And then nonsense variants are also very common in FOXG1 syndrome. And these are small changes. So where one letter or a couple of letters are changed, but they introduce an early stop sign. So if we think about like the body reading the instruction, it hits like an early period and the sentence is stopped earlier than we would expect it to. And so it has a similar consequence as a frame shift in that the FOXG1 protein becomes too short. And so this is another type of truncating variant.
And Ellie, when you're reading the report for a nonsense, when there's a stop codon, so when you gave the example of if you went back to seeing the letters, is that showing exactly where the letter hit the stop sign? So if it were a nonsense, the letters you would see.
Exactly. So the coordinates, depending on what type of variant you have, the letters surrounding the number may be slightly different, but the numbers are going to tell you where in the gene the variant occurred. And so if you had something like a frame shift variant, the tell there is that there will be an FS following the number. FS stands for frame shift. So if you've ever looked at your report and you see that there is an FS in the variant, that means that it's a frame shift variant. And then nonsense variants are either going to have an asterisk at the end, or they'll have the 3 letters TER, T-E-R, or they'll have a letter X. Lots of different ways to say this.
An early termination codon is what the TER is for, I'm pretty sure, right?
Termination,
yep. Yeah, and so Kurt is asking, where does a pathogenic variant fall under these categories? And is it that it's all of them?
That's a really good question. All of these types of changes can be pathogenic. I think it would be very unlikely for there to be a frame shift or nonsense variant that was not pathogenic. We know that there are some missense variants that are normal. So one of the really interesting things about the FOXG1 gene is the vast majority of missense variants, probably more than 99%, all occur within a very specific region of the protein. Missense variants that occur outside that region tend not to be a problem. So it's definitely very interesting. That seems to be an important part of the protein.
Oh yeah, and so Linda just said, is a misspelling the same as a frame shift? So I think
it can be.
But a misspelling is not a missense.
It really can be anything, right? So like we're kind of using misspelling pretty broadly just to describe a change in the sequence. A frame shift needs to have something missing or extra, something that is going to alter the sequence of how we read the letters. So frame shifts will always have something missing or extra, and you'll see that on the reports. A nonsense or a missense variant don't necessarily have missing or extra letters. Sometimes it's just a single letter change. But it's definitely a little confusing.
Right, and it's all being misspelled because that's not the letter that was supposed to be there.
Exactly.
And I'll just take questions as we go as they apply. And Anna Marie has a great question. I think if you go back here, yep. What does the 224 mean? Is it the higher the number, the later change occurred in the gene? That's a great question.
It is. So these numbers actually for every gene start at 1. And so what this is telling us is that, you know, we talked about the DNA example, and then this is the same example in protein. In DNA, we talk about DNA base pairs or DNA letters. And in protein, what we're measuring are amino acids. So amino acids are really the building blocks of protein and what the DNA instruction is coding for. So the DNA is telling us what the sequence of amino acids will be in the protein. And then those amino acids get kind of all folded in on each other to become the mature protein, which then goes off to do its important jobs in the body. So what 224 means is it's counting the amino acid that changed. So in this case, it's saying that the 224th amino acid is different in this person's FOXG1 protein. And then there was a Q&A.
What
Okay.
is the maximum number? That's a great question. So at what point are we safe? You know, is it 40,
FOXG1 is
489?
actually a
Yeah,
relatively small protein,
right.
489.
Is it 489?
Okay, and then there was a Q&A question that was really good too. So Anne wrote, I know you touched on heredity. Do our non-FOXG1 kids have a more likely chance of having a child with FOXG1 syndrome? And that reassuringly, the answer is no. So kind of like we talked about, people who carry FOXG1 variants typically have symptoms. We know that in about 5% of families with a child who has FOXG1 syndrome, when we test one of the parents, we'll find that the genetic variant is there, but it's at such a low level, meaning that it's not present in every cell of the body. It's mosaic. So some of you may have seen the term mosaic on your own reports. And what that's referring to is rather than every cell of our body carrying the FOXG1 variant, only a small percentage of cells do. And it's a small enough number that you wouldn't show any symptoms of the condition. You'd never know that you were carrying that variant. And so the reason why I bring that up is we know that most people who have FOXG1 syndrome, it was de novo, it's not found in either parent. And so those individual siblings are at no increased risk to have a child with FOXG1 syndrome. And even in parents who are found to have mosaic FOXG1 changes, what that typically means is that the variant happened after conception, so after the sperm and the egg came together to form the embryo. And it occurred late enough in embryonic development that not every cell was affected. And so what that means is it's not something that you necessarily inherited from your own parent. And so kind of to bring this full circle, we would not expect parents who are mosaic for their children to be at an increased risk to have FOXG1 syndrome either. I apologize if that was more confusing than helpful, but the take-home message should be that siblings of people with FOXG1 syndrome are not expected to be at greater risk of having an individual with FOXG1 syndrome. Because it's complicated, whenever I get questions about this, I always recommend that, you know, if you have a child who's thinking about starting their own family, it's not a bad idea to meet with a genetic counselor because they'll be able to talk to them about the risk for FOXG1 syndrome, which is very, very low, but also the risk for other inherited conditions that can be passed on as well. And so it's just good to have those conversations kind of earlier, allows people to make kind of more informed choices.
We did have a question from a mom who wrote into me earlier that her son has a 30% mosaic deletion. So if you can explain exactly what that means, a mosaic deletion. I have his genetic code here and I'll put it in the chat. She feels comfortable you talking about it. And she said also what we know in terms of how mosaicism can affect the severity of the disability.
Okay, that's a really good question, and it's one that we're really interested in. So I put this image of mosaicism here because I think it's a little bit complicated. And so this is showing mosaicism, but different ways of being mosaic. So, you know, we kind of talked more broadly that mosaicism in general means that we are not finding the FOXG1 variant in every cell. So if we go back again, if we remember, we have 2 copies of every gene. We have 2 copies of FOXG1. For people who have, like, you know, the traditional FOXG1 syndrome and what we see on the majority of reports that we review, what the lab finds is that when they're measuring FOXG1, in every cell, they're finding that there is 1 healthy copy and 1 copy with the variant. And so if we kind of look at that probabilistically, what that ends up meaning is that when the lab is sequencing FOXG1, they find 50% or half of the FOXG1 sequence is healthy, and then the other 50% carries the variant. So really what they're measuring is 50% variant. Sometimes what we see is that that number is actually lower. And so it might be something like 30% or 25% or 10%. And sometimes it's actually low enough that the lab can't even confidently say what the percent level is. They just know that they saw it, but it doesn't look like it's reaching that 50% mark. And so that's what mosaicism is. It's when not all of the cells are found to have FOXG1. And mosaicism is so interesting. So if we look at this first example here, it's possible to have mosaic changes only in one part of the body. So this is an example showing where all the other cells of the body are completely healthy. But if we look in the brain, a population of brain cells carry perhaps a FOXG1 change. We're not typically sequencing people's brains. We're typically sequencing blood or saliva. So this isn't something that we are measuring frequently, right? This is something that researchers have identified when they're looking at different specimen types. There's also a type of mosaicism called gonadal mosaicism, which is where we can sequence someone's blood and there's no FOXG1 mutation there. But if we were to look at their sperm or their egg cells, we would see FOXG1 mutation there. So that is also a very unique type of mosaicism. And then these other examples are showing kind of more diffuse mosaicism. So rather than it being restricted or localized to a very specific region of the body, what we typically see, at least in sequencing from blood and saliva, is that the mutations are probably distributed throughout populations of cells, and perhaps even more broadly, but it's still not every cell. So in terms of what that means for a phenotype, this is one of our biggest questions. And the reason for that is we know by looking at parents who are mosaic for FOXG1 syndrome that it's possible to carry a FOXG1 variant and have no symptoms. And so what we're trying to understand as part of this natural history study and as part of other efforts that we're leading through the foundation is what is that threshold? So what is like the maximum amount of FOXG1 mutation you can have before you start showing symptoms? Because that's going to be our treatment target, right? How much, like we know we don't need to lower the mutation 100%. What is like our target for reducing the mutation such that we would not expect someone to have symptoms? So that's an active area of interest. I would not be surprised if there are individuals who have mosaicism and FOXG1 syndrome who have milder symptoms than someone who has 50% FOXG1 mutation. And that's just because they still have some cells in the brain producing healthy FOXG1 from both copies of the gene. And again, this is really complicated. So if there are any follow-up questions, I'm happy to
That was a
respond.
great explanation, and I love these images. Do you see where I wrote in the chat with the mosaicism genetics code there? Is
Yep.
there something you can point to that tells us what, sort of like interpret what these letters mean again?
Absolutely. So the variant that we're looking at in the chat, which I think everyone can see,
Yeah.
I'm looking at the C13 del A.
Mm-hmm.
And so this is a great example of a frame shift variant. So do you remember earlier when I was saying, look for FS, that's short form for frame shift. And so we see that here. The C dot stands for coding DNA. So the first part of this is the DNA sequence. So what this is telling us is that at the 613th DNA letter, an A has been deleted. So remember, we talked about for frame shift, there's either going to be something missing or something extra. And so a deleted means that a single base pair, a single letter A at position 613 was deleted. If we move on to the information in the brackets, this is showing the same information, but instead of talking about the DNA sequence, it's talking about the protein sequence. And so what it's saying is that at the 205th amino acid, or the 205th letter in the protein, where we typically see this ASN amino acid, not only has it been changed to a THR amino acid, but it's introduced a frame shift. And when we see this *8, what this means is that 8 amino acids after this new one, there's an early truncation. The protein ends earlier than we would expect it to. And so, very helpfully, we have from Sophia that we think FOXG1 is probably around 489 amino acids in total. And so what we see here is that with this variant, the total FOXG1 protein is only 213 amino acids. So we've lost about half of the FOXG1. So that's kind of decoding this frame shift variant. And then again, it sounds like this is a mosaic finding, so it's not present in every cell. So it's possible that this person may not have the classic description of FOXG1. They may end up falling milder on the spectrum.
Yeah, which I do know that this child walks and presents a lot more like highly autistic. Interesting.
I think that's actually really, I'm grateful for you for sharing that information with everyone, because that's really helpful for us to know that even reducing the mutation by 20%, we see changes in a person's symptoms. And so that's the kind of information that we really want to share with people who are developing therapies for FOXG1 syndrome, because it proves that reducing the mutation can be helpful. Added a variant. And so what I think this probably is N, which is actually a different way of saying ASN. So it's the same variant or the same amino acid in the previous example. It's just a different way of saying it. And then 253, and we wrote 1 here, but I'm almost positive that that's probably an I. And so this is going to be a missense variant, which is where one amino acid has been changed to another. So the length of the protein hasn't actually changed at all. There's just a small difference. So in this example, if we look at the C dot, we remember C dot means DNA FOXG1. What we're saying here is that at the 758th letter of the FOXG1 gene, an A has been substituted for a T. And what that means for the protein FOXG1 is that at the 250